All right, everyone, this is the initial first lecture for Humoral Immunology. Now, if you'll notice, the textbook we're using is Janeway's Immunology, or in some places you may see it referred to as Janeway's Immunobiology. Tomato, tomato. One of the things I also would like to point out to you is that this book is copyrighted 2012. So while it is a little out of date for what we're doing, it will suffice. Um, this will give us a good broad background in the field of immunology, and for the more up-to-date research and findings, we will be going to the primary literature. Now, as in everything, it all has to start in the beginning. When we trace back immunology, we trace it back to, literally, Edward Jenner and his vaccinations. Now, I'm sure many of you have heard the story of Edward Jenner and his research into smallpox. For those of you who haven't heard the story, the urban legend is that during this time period, I believe it was mid-1700s, there was a bit of a smallpox outbreak, which is a bit of a misnomer. Everyone got exposed to it, everyone got it, but this year, for this time period, it seemed to be an extremely hard time for this. During this time, everyone was getting exposed, and a lot of people were dying. Edward Jenner was one of the few people to notice that while everybody was getting sick, from the aristocracy on down to the street urchins, everyone was getting exposed, everyone was getting sick, except for a small subportion of the population, the milkmaids. Now, generally, most people in polite society didn't even notice or acknowledge the fact that there were milkmaids, because think about it, their job was to be out in the barn, ankle deep in you-know-what, milking the cows. They would be filthy, because working in a barn with a bunch of cattle is not a clean job to begin with. So, ankle deep in the muck, getting rubbed by these cows. And it turns out cows have their own version of smallpox. We refer to it as cowpox. Has some of the same symptoms. Sores that burst open become a weeping sores, but with the cows it wasn't debilitating. They would, given enough time, those sores would heal and they would be fine. For us, the smallpox could be debilitating, and if you're lucky, it just left a lot of scarring on your skin, lining of your sinuses, your mouth, your airways. Jenner noticed, though, that, like I said, these milkmaids were not getting sick. When he looked at and he saw what was going on, he noticed that they were rubbing on the cows as they were trying to get the cows to move, and as cows being these, half-ton behemoths, didn't care what a little 100-pound person wants, would, walk all over them. So these milkmaids were getting rubbed on with these cow poxes, these sores. The newer milkmaids would, inevitably get rubbed on by these sores. They would break out in a few days later with some small pustules. Pustules would burst, they would weep, and then a few days later they would heal up, maybe scar, maybe not, and that was the end of it. They would never get sick again with the cowpox, and miraculously they never got sick with smallpox. So, Jenner had an idea. He squeezed a bunch of these new pustules on cows, collected the pus, and then basically started vaccinating. Grabbed a needle, dipped it in the pus, started jabbing people, volunteers. He thought he had a way to keep people from getting sick with the smallpox. Lo and behold, he did. Came up with the first vaccination. Turns out vaccination is named, the word is termed after the virus for smallpox, vaccinia. And that's where we get it. Fast forward about 100 years, you get to Robert Koch. Robert Koch came up with his 4 postulates, are ways of trying to show, to prove that a specific organism is responsible for a set disease. It wasn't their humors were off, it wasn't the cold, it was a specific microorganism. Followed by his contemporary Louis Pasteur. We all know Pasteur. Pasteurization was able to show that microorganisms cause spoilage of food. But what we don't always come to mind with, first of all, is the fact that Pasteur himself was also into studying disease processes. He came up with vaccines for many different viral infections. And then we have Behring and Casanto in the 1900s came up with the idea leading to what we now think of as antibodies. Serum proteins that float around in your blood that will bind to and attempt to protect your body from getting infected. Now, we got to keep in mind that our studies of many of these things are not in vain. When you look at things such as the smallpox vaccine is one of the first times that we humans actively set out to reduce human suffering due to disease. When you look at the number of cases in the 60s, yes, it says only less than 30 countries had smallpox cases, but you got to remember this is thanks in part due to modern day hospitalization, modern day treatment, modern day water treatment, and all these ways of trying to reduce disease being transferred person to person. 60s into the 70s, the CDC, the World Health Organization, the United Nations, and many other countries, backed with the U.S., went literally to war against smallpox. They went out and they tried to vaccinate every person possible. Now, does this mean they were able to literally vaccinate 100% of the world population? No. This is one of those times where all you have to do is get enough. Herd immunity will take over. If a large enough portion of the herd is vaccinated or is immune to an infection, once the people that have the infection get over it, there's no place for that disease to spread to. Herds now clear. Last reported case, I believe, was 1976 or so. And then 1979, we declared smallpox officially eradicated. There were no known infections in the human population. Now, when you go and you look at the immune response, our immune systems have evolved to take on any infections we may be exposed to from different prongs of attack. We have 2 of the prongs known as adaptive and innate. Adaptive, anything that adapts, learns, it overcomes. So this adaptive learned immune response, these are going to be cells that are there lifelong. They are learning new things. They are continually adapting to new things you may be exposed to. And once they've been exposed, once they've learned this is something that is bad, something not you, they will remember it for your entire life. As long as that cell line is there, there will be memory. The innate, well, this is non-specific. This is not learned. This is something that's immediately available. These are going to be serum proteins, proteins like the antibodies I just mentioned, and other proteins that we'll be talking about like complement. These will bind to anything not you. Doesn't matter if it's bacteria, if it's virus, if it's parasite, they will go. And then lastly, there are cells. Cells that are constantly on patrol. Anything not you, anything not living in you, they will phagocytize, they will eat, they will destroy. One of the terms you're going to be hearing a lot throughout this course is what is referred to as antigens. Antigens are anything that elicits a response, stimulates antibody generation. Antigens comes from antigen. These are going to be any types of proteins, glycoproteins, and polysaccharides that are going to stimulate B cells. B cells are the cells that are responsible for making the antibodies. Antibodies that will bind to and will opsonize, coat anything that's trying to infect you. Now, these are also some of the things that can be considered allergic. Certain metals, drugs, and other organic chemicals. Remember, organic here means carbon-based. So if you're allergic to penicillin, you're allergic to strawberries, it means something that is not truly an antigen is provoking an immune response. Now, everything begins in the bone marrow. Pluripotent hematopoietic, not even going to try to say that word, ladies and gentlemen. Hematopoietic stem cells. These are the blank slates. They haven't been told anything yet. They are waiting to become. They are waiting to be told, you will be this. Everything here is going to be based on recognition, regulation, and memory. How does that pluripotent hematopoietic stem cell know what to become in the bone marrow? Well, it's going to receive signals from cells around it. Well, how's it going to know to continue to differentiate, to become something different? Well, more cells, more signals. The more signals, the longer they last, the more it will develop. The same signals, some of the same signals that told it to develop to become a mast cell, to become a T cell, will also tell it to stay alive. Stay alive and to increase in numbers. We see that we start with these blank slates. The blank slate is kind of told, hey, you're going to become lymphoid, or you're going to become myeloid. The lymphoid progenitors are going to become our B cells, our T cells, our NK cells. These are going to be the guys that are responsible for our adaptive immune response. B cells will become plasma cells, which will then make antibodies. T cells will then become activated T cells that are responsible for hunting down and killing virus-infected cells. NK cells, natural killers, they go and kill anything that is infected or anything not you. The myeloid cells, these are going to become what we think of as the bone marrow cells. They're going to become the white blood cells or the red blood cells. As they are told, and here remember, being told is to be exposed to new chemical signals. As more chemical signals show up, the progenitors will become the white blood cells. Some of the white blood cells, the monocytes, will become the macrophages or the mast cells. These are the cells that are constantly on patrol, going throughout your body, looking for anything not you. When you look at the different cells. Each of the cells has a job. Each of the cells has a different role. And if you've ever had me for a different biology course, you know I always say shape dictates function. Well, if you look at the different shapes, you will see the different shapes denote or the macrophage. No, the eosinophils will just disgorge all of their contents, all of their digestive enzymes onto the surface of the worm. Break down its cuticle. Break down, allow for other cells to get in to the interior of the worm and begin doing damage. Basophils. Another one that is great for anti-parasitic. Each of the big granules you see there are large proteins, stuffed, enzyme-packed vesicles waiting to be disgorged onto the surface of something that is too big to be phagocytized. They will break it down and then allow for macrophages and other things to come in and clear it out. Turns out basophils are also part of the reason why many people have allergies, hay fever, things like that, is because the basophils and the mast cells we'll talk about shortly, they will respond to something that is not an antigen. They will mistake it, and then they will over-respond, which causes things like overproduction of the mucus in the sinuses, and now you have a stuffed nose. Whole point of mast cells is not for, killing off of anything, not for presentation of anything, but it is for amping up a response. All the cells we've seen will respond to different signals, different cytokines being chemicals that go up like flares that say, hey, here, over here, there's an infection, come here. Well, the mast cell just takes that and then turns it up to 10. Maximum effort, maximum response. So when you go and you look at many of these cells, the innate immunity, this is our first line defense. Anything that can get through the skin, through and onto and into the mucous membranes, these are what it runs into first. Secondary defenses are going to be the B cells and the T cells. These are going to have to be activated by the dendritic. The dendritic, like I said, is the server that takes antigens, takes things from the granulocytes, and then delivers them to the B cells and the T cells. Now, our friend, the natural killer cell, recognizes cells that are abnormal. A cell gets infected with a virus. Remember, one goes in for viruses, thousands come out. Well, it changes the cell. It changes how it acts. It changes its overall plasma membrane, what's presented on its surface. Natural killer cells recognize this fact. Natural killer cells will then go and kill any cell that is acting abnormally. Because think about it. Better to lose the 1 in a trillion cell that may or may not be viral infected than have a virus spread through that tissue, loss of the tissue, loss of the organ, loss of the organism. Now, our friends, the effector lymphocytes, as they're referred to. These are going to be the B cells and the T cells. Generally, they sit there in different places throughout the body. They are naive. They have not been exposed to anything. They've gone through a selection process that we'll discuss in a later chapter. So that way we know they will not respond to self, respond to your own tissues. But now they're waiting. They're like all queued up, waiting to be called. And as soon as they are called, the B cells, once activated, will become what are referred to as plasma cells. These are going to be cells that all they do now is produce antibodies. They will flood the bloodstream with these plasma proteins. The T cells, once they become activated, will go and they will hunt down any cell that is viral infected. Any cell that has a bacterial infection in it that it cannot kill. T cells come in numerous flavors. All T cells have what is referred to as a T cell receptor. This is a protein complex on the surface that literally acts as a switch. If the correct antigen can bind to the switch, T cells turned on. Until that antigen binds to the T cell receptor, that T cell now will be quiet, will be what they refer to as quiescent. It will sit there and wait. Now, depending upon what other proteins are on the surface of the T cell, you can have either what is referred to as a cytotoxic T cell. This is the one that hunts down, kills infected cells. A helper T cell that literally does as the name says, it helps. It provides chemical signals, chemokines. Chemokines here are going to be an attractor. Floods a tissue with these chemokines. It tells other cells to home in to the site of the infection. And then regulatory T cells. Everything that gets turned on in the immune system must at some point be turned off, suppressed. That's what regulatory T cells are for. Now, once activated, doesn't matter which of these three types of T cells that cell will become. It will have a long memory, because once that cell gets activated, as we see in a later chapter, it will divide. 1 becomes 2, 4, 6, blah, 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 and on and on. Once the regulatory T cell decides it's time to suppress activity, well, most of, but not all, those T cells will die off. That way, what started off as one cell after the infection, maybe a few dozen, sitting around throughout the body, waiting till the next time you get exposed. Peripheral. Everything originates in the bone marrow. Once it hits a certain stage in its development, it'll then go out to the bloodstream. Passes through a few stages of development, then it goes to the lymphatic system. And that's where it will reside. In the lymphatic system or out in the bloodstream, circulating back and forth. End of first lecture.